Hello there, AP Bio. I've got another FRQ walkthrough for you. This one is now part 8. It's a fresh start, so if you haven't done 1 through 7, that's fine. Part 8 is a brand new beginning of a question. I'm going to be looking at the 2020 practice exam number 3, question number 1. So here we go. This question is, you can find it in the AP classroom, but it covers chemistry of life and some enzyme energetic stuff. So some mitochondria content, how enzymes work and things like that. I say a lot of ums, sorry. This is about parasitic cells. They're eukaryotic. So they are multi-organellular, like they've got organelles and a nucleus, but they do not have a functional mitochondria. So they get into your digestive system and cause illness, and they are getting their ATP just through glycolysis, but they don't have a Krebs cycle and an electron transport chain. So they finish out and kind of like create a loop by doing fermentation with this enzyme, lactate dehydrogenase. So researchers are using 2 chemicals that are non-competitive inhibitors and to that enzyme. And then you have graphs of what they call, where does it say? Relative investigate concentrations. They have graphs of relative growth. So as concentration of the chemical increases, here's the relative growth of this experimental group with Gossypol or FX11 or FX2. And relative growth means that there is another group that we are comparing against. So at the beginning with 0 concentration, 100% growth without the chemical, 100% growth with the chemical because there's none added yet, right? 0 concentration. But as you up the concentration, you're getting like less and less growth here. And that's why this graph is going down. So here are the prompts, A, B, C, D. This is a question number 1 style FRQ, so you'll be timed about 25 minutes for this one. So if you want to, before you move on in the video or before going through the PowerPoint, try it out. This is where the link will be. And here we go. Let's see how fast I can do it. So in A is typically, if I go to the very beginning, in a style number 1 FRQ, A is describing and explaining concepts about biology. It's just content regurgitation being applied to this scenario. So that's one that it's good to like maybe have your notes with, double check, and like usually you guys know this stuff by now, you're probably going to get it. B is going to be something about the experiment, controls, independent, dependent variable. C is going to be something about looking at the data of the experiment specifically. And then D is going to be like something is changing, make a prediction, and justify with content again while you're doing that. Okay, so let's look at what they ask us just about content on this one. All right, so how C. parvum, describe how C. parvum obtains the glucose it needs for glycolysis after it has infected another cell. Explain the role of lactate dehydrogenase in enabling this cell to continue producing ATP by glycolysis. So you have to take what we know about cellular respiration with glycolysis, Krebs cycle, etc. But if they can't do the mitochondrial parts, like they're doing glycolysis into fermentation so that they can get their electron carriers emptied and still go back into glycolysis and generate some ATP. So this question, don't overthink it. It's basically asking, where does the glucose come from? You can't create it out of nowhere. And what is that enzyme's role, or what is it doing in this pathway or in this process for these cells? So for number 1, where is the glucose coming from? It's absorbing it from its environment, taking it away from the host. You could say something like it's coming from the digestive system of the host, because those are the cells it's infecting. And for part 2 here, explain the role of the enzyme. If it doesn't have mitochondria, it needs to empty its electron carriers somehow, right? In glycolysis, you're oxidizing, losing electrons, oxidizing, Leo, right? taking electrons that are excited away from glucoses, putting them on an electron carrier, and normally you'd go to the ETC, but you don't have that in these cells. So they go to fermentation, offload the electrons with the help of this lactate dehydrogenase, and now that's NAD+, it can go back to glycolysis and do it again, right? And it's getting the ATP just from the glycolysis part. So their answer, this is where you do have to have like some specific content. Please don't use like shuttles or trolleys like we've been using for these electron carriers, but they're actual names. So that's a good time to have your diagram out. If you've been looking through these, I have former pictures of the diagram back up in here. So, but find yours, you want that. Let's see, what does it say? Lactate dehydrogenase allows fermentation to convert NADH, the reduced electron carrier, right? Gained electrons reduced for GER, Leo the lion goes GER. So taking it from that form to NAD+, plopping off electrons in fermentation. I don't know what they do with them. Maybe it makes alcohol, maybe it makes something else. Probably lactate, don't know. And then if you've lost electrons, you are now the oxidized form, which is needed for glycolysis, and it goes back making more ATP. So that's A. B, this is about scientific process stuff, about the experiment, okay? Identify the independent variable used in the experiment. So if you look at the graph, make this bigger. This graph is pretty straightforward, right? X-axis is actually our independent variable, Y-axis is the dependent, and in this case it's the concentration, you could say of Gaussy-Paul FX2, or you could say of the chemicals, right? So you don't have to get specific. Move that off to the side here. But in this case, independent variable is concentration of the chemical. Don't just say the X-axis, or it is the or variable on the X-axis, because if you've looked at former FRQs in here, like sometimes they're different depending upon the way it's graphed. The reader doesn't know, who's grading your FRQ, that you know what an X-axis is. Is it this one? Is it this one? So you have to be specific and state the variable, okay? B, second thing, this one's 3 points. Identify the difference between the control cells and the experimental cells used in this experiment. So you just have this one set of graphs that has two sets of concentration, right? But you don't see anything about control cells. So from the text, they did tell you, in experiment, human cells were treated with different concentrations as the experimental groups of the Gaussy-Paul or FX2 after infection with this eukaryotic parasite cell. And the relative growth, I'm going to underline that, of C. parvum compared to that of control cells was measured. So when you're looking at relative growth, it's a comparison of the one that you're observing, right, with the chemical treatment compared to the one that's growing just fine without the chemical. So that's the relative growth. You can see that they both start at 100, the experimental group here starts at 100 when there's no chemical added. So that's like the control. They're going to keep that no chemical added as a control. And then if you add 10, well, no effect really. If you add 20 concentration, we're dropping. 30, we're dropping, right? And so it's the addition of that chemical in the experimental groups, but to your control group, that would have been no chemical added whatsoever. So the control cells were the same types of cells infected with the parasite. So this is human cell, like our intestinal tract. They're not getting real specific here of tracking that. That's okay. But not treated with any chemicals, as were the experimental cells. Remember, the experimental groups treated with the chemicals to see what happens, control cells not treated with the chemicals. And the third one, justify the researchers using a different range of concentrations for FX2 than was used for Gaussy-Paul. I love that word, Gaussy-Paul. It's a lot of fun to say. It's unlike the other ones I haven't been able to pronounce. So look closely at these graphs, right? Gaussy-Paul is going from 0 up to 40 micromolar, and FX2 is going from 0 up to 80. The graphs have the same shape, but the range of concentration is different. why would I keep upping the concentration of Gossipol if the cells are already 0% growth, right? Dying after about 35-ish here. Why would I stop treatment at 40 on FX2 if I'm only cutting the growth down by 40% relative to a control group? I need to keep upping to get the same results. So that's what they're asking you for in this third part. So you just have to look closely at the data on the graph and kind of have a justified, like, why are these graphs not the same? Why don't they have the same axes? So they said FX2 had a minimal impact on the growth of the cells until it reached a concentration of greater than the concentration of Gossipol that was tested at 30 micromolar, which was the maximum. This is really wordy. I think a lot of answers would probably satisfy here as long as you are making sense, comparing the two. Why was this one different concentration than for, yeah, they're not looking for like one, only talk about one chemical and not the other. They're talk about both. So yeah, let me know if you have questions on this one. I think it's straightforward. I'm just coming up with a blank right now. All right, so A is about biology content, B is about experimental design, C is usually about analyzing data. So this one is a one-pointer here. Based on the figure, identify the concentration of Gossipol that reduced C. parvum growth to 50% of that in controls, 50% of the control cell growth, right? Control cell growth has 100%, Gossipol took it down to 50. At what concentration of Gossipol? So you got to look at your graph here, and you're looking at 50% growth on the Gossipol graph. This is all you got to do is just read the graph, right? Not too tricky, but you easily, on your FRQs in my class, you guys screwed them up. So take your time here. The graphing questions are not hard. Just go slow and double check yourself, okay? So like underline and like observe what you're reading here. So I'm looking at this graph because it's for Gossipol. I'm looking at 50% concentration. I don't have a line there. So if I want to get my clear edge ruler out and put it on my computer screen, right, so that I can tell and read accurately across, do that, okay? Or even a straight edge, like a piece of paper. And then that's this data point. That's the 50% growth. Well, what concentration of Gossipol did that? And it's right in between 10 and 20, so it's 15 micromolar. And this answer did have the units written out. I know micro might be a weird thing if you're typing in Word. You could just also write out micromolar, right? If you can't get that like symbol, because M is millimolar, not mu micromolar, okay? I don't have enough time, 2 minutes left, to do part D. So that will be in part 9. Okay, so this is part 8. And I did A, B, and C of practice test number 3, question number 1. All right, thank you guys.